of neutrophils in the immune system relates to sort of neutrophils having most abundant white blood cells in the body's sort of first line of defense to against infection. And their functions include phagocytosis, degranulation, and the release of neutrophil extracellular traps, which collectively sort of try and support in combating or invading pathogens. Whereas if we look at sort of neutrophils in healthy donor normal response, normally we see that neutrophils have a very different role. So they help protect us from infections. However, as we'll see in the context of severe COVID-19 patients in a very specific cohort where they collected both mild, sort of severe and also fatal COVID-19, we'll see that sort of this dysregulation has sort of different forms of sort of protective functions, and it can also lead to more harm than good when you have these sort of neutrophils that are dysregulated. Okay, so a bit of background on sort of the application of neutrophils, particularly in the context of severe COVID-19. So in the immune system, normally when it comes to sort of the pathology of severe COVID-19, sort of these neutrophils become hyperactivated, resulting in a sort of cytokine storm. And how we can see this is that neutrophils play a major role in the overreaction of the system. And during the cytokine storm, we can see that these neutrophils produce high levels of reactive oxygen species, and they release very specific cytotoxic enzymes like meloperoxidase and elastase. And although these molecules are intended to fight the virus, there are sort of forms of excessive production, which leads to significant tissue damage, especially when it comes in the context of particular, areas of the lung, and contributing to acute respiratory distress syndrome in these patients as a result. So we can see here in the diagram sort of the different differentiation of these different neutrophils in different populations, how they relate to sort of blood, and how these contexts relates to sort of tissue damage when we see this release of the cytotoxic enzymes in the bronchoalveolar space. Okay, so now we'll try to really understand what's the role of this sort of enzyme arginase 1 in the context of these patients with severe COVID-19. So arginase 1 as an enzyme is known to modulate inflammation by controlling the availability of arginase, which is a key sort of molecule in immune responses in the body. And in a healthy sort of system, if we sort of look at the context of arginase 1, this sort of enzyme helps reduce inflammation once an infection is under control. However, when it comes to severe COVID-19, arginase 1 becomes dysregulated, particularly in the context of neutrophils and how it's expressed within different neutrophil populations, which in turn fails to properly release sort of the enzyme. And this results in sort of a prolonged inflammation, which worsens when it comes to tissue damage, especially again in the context of different areas of the lung. And we can see sort of these results here in terms of what types of populations of neutrophils and how this sort of changes as well. Okay, so what is sort of, again, the focus that we'll try to sort of uncover from this sort of topic? So we'll try to sort of dive deeper into this neutrophil dysfunction. So normally most of the studies, they do try to analyze the context or the role of arginase 1 in neutrophils. But there's not really studies that try to really, at a granular level, try to subset specific neutrophils with positive expression of arginase 1 and those that are negative with arginase 1. In order to be able to then go further into how these could relate to outcomes in these patients, as well as differences in how these neutrophils sort of differentiate. So I guess the context of this is that we will be utilizing the Talk to Data repository system. And again, for any scientists or any sort of researchers who are joining sort of this session for the first time, and they're not actual users of Biotrain, so we'll use sort of a specific application in sort of the Biotrain ecosystem, which is our main repository where we hold all of the publicly available source data, whether it's single-cell RNA-seq, single nuclei, TCR sequencing, or spatial transcriptomics technology or imaging data. So that's where we can harness the public data to really look at the expression of this gene and to see if this relates across different studies, and particularly in the neutrophil population, and what exact subset of neutrophils we're seeing this sort of dysregulation. Okay, and we'll then move forward into going into a particular study, looking at sort of severe COVID-19 compared to mild forms of disease, of COVID-19, and if we see any differences relating to sort of this neutrophil subpopulation or this sort of expression of arginase 1. Okay, so in order to do this, we'll have sort of two aims that we'll try to sort of understand. So the first one will be sort of using the talk to data system to be able to uncover transcriptional signatures of neutrophil dysfunction in severe COVID-19 by focusing in sort of this population with arginase 1 dysregulation and its impact in neutrophil maturation. The second step of sort of the analysis that I'll show you how to perform on the system is to look at a specific spatial data set that also relates to severe COVID-19 patients and be able to map the spatial distribution of these sort of dysfunctional neutrophils by looking at the expression of arginase 1 in the lung and particular different areas in the lung as well, because spatial data usually has different morphology regions, different pathology areas, and we'll try to see if we can actually find any sort of expression or co-regulation of certain cytokine inflammatory markers as well, where we see this expression of arginase 1 in the lung. Okay, so these are our sort of aims for the session today. So in order to do this, we'll sort of go into the talk to data system first, so I can show you how you can get a bit more understanding on how certain targets are expressed in the context of different cells, different tissue level data, and different conditions. So in order to be able to sort of map this information before you go into a specific study analysis or selecting a specific study that might have data relating to your area, you can go into the talk to data system and you'll see that there are two layers in the talk to data system. One, which is the former version of talk to data, which you can actually use to find particular studies and filter based on certain sort of standardized metadata labels. And our new metadata, sort of our new beta version of the talk to data system, which is built in a different way. And it allows you to really map target expression and be able to create a sort of tree ontology of different sort of labels for that ontology system. So if we go into the new beta version, which is going to help me get a bit more context into sort of severe COVID-19, different sort of maturity of different neutrophils and how arginase 1 is expressed. And this is, again, in the context of all single cell RNA-seq data in the database. So I'll be able to actually filter certain sort of labels and create my ontology tree with different roots and different sibling sort of root cells or labels. So on here, the first step that you'll need to see is either you can create a new ontology tree where you're creating the root node and you're adding those sibling nodes with different layers of the data, or you can use our BT suggested ontology or your own sort of default ontology system. So the first step here is to really look at different neutrophil populations across a database. So the first step is to query these different neutrophils. So I can actually gather sort of some information on differences from mature and immature neutrophils. Okay. Once we have those labels here, we can then move forward into adding other information. So the next step is going into the Mondo disease ontology system, and this will help us sort of gather information just relating to COVID-19. So we can add that sort of label here. Our next step is to really understand how this enzyme is sort of expressed in the context of the sampling site or the sort of sampling site of those samples in those studies. So it could be different where it might be expressed in particular areas in the lung or in sort of an injury or a sort of tissue damage region, or it might be that it's actually more enriched in blood samples and could be used as a potential biomarker as well. So in order to really be able to get that information, you can choose the sampling site as well. And this is where you'll be able to select different nodes relating to different sampling sites in the data. And I'll select here the disease involvement site. So this could relate to sort of areas in the lung that again have some form of tissue damage. I'll select samples relating to just blood from these studies and also fluid, which could be bronchoalveolar lavage or other sort of forms of sputum that are collected for these studies. So once we have all this information and our specific filters, we can then go forward, and the first step to do is to query our particular sort of target gene so we can actually see how this enzyme is expressed in the context of these layers of the data. When we search, you'll see that there's some parameters at the bottom that you can sort of change here. And I'll zoom in a bit closer. So I want to, instead of making the scale absolute, I'll change it to a relative scale. And I can actually see if you're mapping it on a larger sort of scale, so a lot of different nodes, not just three, and you want to be able to see it a bit clearer, you can also change the average rank or sort of limit to a specific intensity as well. And this might be specific if you really want to uncover specific targets that have a high enrichment in certain tissues, and you can sort of label that or change the average rank to be able to locate those specific nodes that have that information. So once we're here, what you can do in Talk to Data in the new beta version is go into the heat map. And what I want to do is specifically go in mapping different levels of the data to see those differences. So one is sort of the neutrophil population. I want to see if there's any specific differences. The other part is sort of the disease. So you can again leave it, you can add disease first since it's easier to just see one nod. And then from there, you can bring the differences in the different neutrophil populations. And you can then add the third layer, which is looking at different sampling sites. So what can we gather from this? So from all the data sets in the system that are relating to our specific ontology tree or our filters, our labels, we can see that within data sets relating to COVID-19, and again, across different severity levels, mainly arginase 1 as an enzyme is expressed within immature neutrophils, this particular population. And we can also see that it's mainly sort of found in blood samples. So we don't really see arginase 1 expressed in bronchial lavage, sort of fluid samples or other sputum samples in the lung that are collected. And it's also not as enriched in the disease involvement site. So we don't really see a lot of papers or a lot of data sets that are trying to really understand the expression, which is also a reason it could be low, or it could just be how the study subsetted those populations of neutrophils and analyzed it in the context of the samples. But we can see here just from this information, we can gather that it's mainly in immature neutrophils where we're seeing that expression. So this is where you can, again, gather a bit more information on the number of studies, the average rank, and also more about that population of neutrophils and the percentage of cells that are sort of expressing arginase 1 in the database. So this is the new system where, again, you can create your own ontology tree that is specific to like a cancer. You can create it based on fibrosis, on a particular condition, and add your sibling nodes to be able to understand a lot more sort of clinical data parameters or other information you want to gather. Okay, so if we go back, this is what I was showing you how you can perform this analysis in the new beta version. The firmware version of Talk to Data, which is something some scientists might have already used if you're a current user of Biotrain, is being able to actually locate what data sets we have in the system that particularly have the condition, so samples relating to COVID-19. In this case, we can filter only by neutrophils, so the system will gather only those cells relating to different neutrophil populations. And then we can also ask the system to only provide us cells that have a positive expression of arginase 1. It's not a particular intensity, so that's why it's a bit different from the new beta version, but it's just showing us what data sets can have this relatedness of this arginase 1 positive expression. So you can do this as well in the former version if you really are interested in interrogating all these specific studies, and especially if you want to integrate the data sets into an atlas. So if you are working on generating very specific custom data set or specific atlases, this is where you would be able to generate that create custom data set where you would be able to pull in all these filtered cells based on your labels that you've added and be able to then run very specific downstream analysis. So this could be differential gene expression across different samples in the studies. This could be pathway enrichment analysis or even monoclonal 3 trajectory analysis across all these different neutrophils from these 17 data sets in this case. So this is sort of the two ways you can get an idea of what public data we have. And in this case, we'll be mainly focusing on this GCASC 174072 data set, which particularly has a lot of information relating to neutrophils, so a lot of cells in that cluster. And the sort of study looked at different forms of severity. So we can actually use that data to look at differences in mild versus severe, especially when we're trying to subset only those neutrophils with this arginase 1 positive expression. Okay. So really, in order to do that, we'll need to then go into the bBrowser X system. So I'll remove this talk to data and we'll go into sort of the data set that we sort of found from the talk to data system. So normally when you open up any public data set, you'll be able to visualize that study and a bit more information on that data set in the system. And you'll be able to see all of the metadata associated with that sort of study. So that includes any clinical data, any parameters that you want to run further interrogation on or analysis on. Or it could be that you just want to visualize what are the author cell type annotations that were defined in the study or other information relating to sort of severity as well. So you should be able to see this in the dropdown. And in order to be able to then subset my specific population and be able to run differential gene expression or pathway enrichment, the first step we would need to do is to subset only these COVID-19 samples. So you will find that many data sets in the system include studies where they have a control, so healthy donor samples and also different disease sort of condition subgroups. So in order to do that, we have to make sure that we're only selecting all of the COVID-19 and we can then perform subclustering. So subclustering, what it does, it isolates that population so that any downstream features that you're applying or any analysis that you'll run, it'll only be applied to these cells that you've subclustered. And this sort of feature is quite useful because normally you would have to have coded this on R or Python. It takes a bit longer to do. You would need to have the experience on how to actually run the script to be able to go into that granularity. From there, if you want to only concentrate on neutrophils within these COVID-19 samples, you would go into sort of the author cell subtype, or in this case, it could be your own clusters or your own annotations. And this is where we're going to select our neutrophil population. So we can see there's over 15,000 neutrophils in this data set, and we can perform a second level subclustering. So that will only be COVID-19 neutrophils within the study. And from here, this is where we can actually look at arginase 1 expression. So we're able to actually subset only those neutrophils that are positive for that enzyme. So if we add here arginase 1, we can then search for the expression intensity. So we can see it is expressed within these neutrophils. And the way that you would be able to see whether it's expressed or not expressed to create that label for those cells is to change that unit to a smooth binary. So instead of having a normalized intensity of that expression of that gene across different neutrophils, you can change to the smooth binary. And that would give you cells that are both expressed within these samples and those cells that have a negative arginase 1 expression. So if we're interested in this population specifically, the step that you would need to do to subset and actually label these cells is to go into the label add annotation feature. And this is where you can add your own population. So this is sort of COVID-19. And particularly it's relating to our neutrophils, and this is arginase 1 positive expression. Okay. And again, I'll just add a secondary label so it adds the positive group. And actually I'll change this just so it's arginase expression. And that's when I can add this group here. Okay, so once I have my metadata labels, I can add that group into the system. And you'll see that it now generated my own metadata where I can specifically see my cells that are positive for this arginase 1 marker. And these unassigned cells are all the other cells that were negative for arginase 1. So if you do need to label these to be able to have both groups, you can go back to label. And there's a way to easily append any sort of new label to an existing group. So for example, I had my COVID-19 group that I just created. So I'm going to append this new label for the unassigned cells, and I'm going to add arginase 1 negative so that the system knows that these cells are negative for that expression profile. Okay, so once you have this, now you have your population, and you can then subset only these arginase 1 positive cells. So even go even further into subclustering the cells only. So again, I can go back to my label here that I had before. I'll select my positive arginase 1 cells and I'll subcluster these cells even further. Okay, so once I have my population, this is what I was showing here in the slides, where you can look at different expression levels and look at differences in different severity scores as well. So the next step is to be able to perform differential gene expression, where I'm trying to see how these neutrophils are activated in the context of severe patients versus mild forms of disease. So in order to do that, we'll again remove the arginase expression and we'll go into this differential gene expression feature. So this will allow us to be able to create a design for the DGE analysis or study specific design question. And you can map different forms of the data. So we have our subclustered arginase positive neutrophil population, and I want to be able to map from this population the different severity levels as well. So normally here, you'll have your metadata that you generated before, so you can actually see your label, and you can add any other labels relating to the actual severity. So any study would have this information, and it's just a fact of just adding it and being able to then generate the score. So you can then add specific groups that you want to compare in the differential gene expression analysis. So I'll add here my group because I actually combine all severe and all mild into one. So I use the same labeling concept to just create the sort of integrated format. And I'll go into my 3 WHO scale and generate the chart with my label specifically. So here I have my mild group. So in group 1, when you're comparing this, you'll need to add the label so that when it runs the DG analysis, you know what genes are coming from which group. So just make sure that you add that information before doing anything to the analysis. And I'll add mild disease. Okay. And this is arginase 1 positive expression. So this will be my cells in the mild group. My second group could again be the severe group. Okay. And then arginase 1 positive. Okay. And I'll add my severe group here. So once I have these cells sort of identified and I have the design, I can include or exclude certain genes as well. So there might be cases to your case or your data set that you're using as an example that you might want to include certain gene sets to be able to see if those target genes that you're trying to sort of identify or understand further, if they're differentially expressed across certain treatment groups. So you can actually include those gene sets specifically, or you can exclude certain genes that might have a high mitochondrial percentage and it might create noise when you're trying to sort of understand the analysis. So there's different ways on how to adjust those parameters and then what statistical tests you're going to use to perform that DE analysis. So in this case, I'll leave it as Venice, which is one of our main methods when we're looking at single cell RNA-seq data, and we'll run the analysis here. So I do have a sort of example because it does take a few minutes to load all of the features. So when we go into the analysis, we can then see that there's very different expression profiles compared to severe versus the mild forms of disease, particularly in our arginase positive neutrophil subgroup that we created. And we can actually identify very specific markers as well. So these particular markers, FCGR3b, RNA18S5, I can show you sort of some information on this. So we can see that there's some form of downregulation of these neutrophils that are activated genes in severe patients. So CD16b, CD16a are particular genes that are downregulated, which indicates that there's a dysfunctional activation in arginase positive neutrophils. And we can see this based on other literature that has been published already, where we can find these similarity in these sort of concepts where we can see hyperactivation of these neutrophils in only forms of severe COVID-19. And we can also sort of understand that there are some dysfunctional neutrophils that lead to ineffective immune response modulation. So we have these particular markers here. We can also see that there's also marker ILR2 or IL1R2 in the list, which is a marker known to modulate inflammatory responses. So that's also the case where this particular marker is found mainly in mild forms of disease. So it's able to modulate that inflammatory response, and particularly within this group. So as well as if it contributes to the cytokine storm when it comes to COVID-19. So it has two different roles as well within the analysis. Okay, other sort of genes that you saw from the list as well is these ribosomal RNA components in severe COVID-19. So we can actually also identify that these particular RNA markers are downregulated and are reflected of sort of reduced protein synthesis and activity in these arginase 1 positive neutrophils, and particularly only in the case of severe forms of disease, so not the mild forms of disease as well. So in this sense, you can sort of get this context that this sort of information relates to sort of exacerbation or the dysfunctionality of these neutrophils within this case. Okay. So again, a summary that we can gather from this is that we were able to tell that these arginase 1 positive neutrophils do have a dysfunction, and they do have enhanced dysregularity in severe cases due to the sort of defects in this arginase 1 release and altered gene expression. And this finding does align to literature, which we showed different literature concepts that were previously sort of discovered from other different studies, and how these sort of dysregulation can contribute both to increased tissue damage in the lung and poor disease outcomes in COVID-19. So I guess from here, what is the step, or what can we even gather further from this to really understand the context of these markers? So what we can do from the DG analysis is actually look at different pathways that are enriched compared to mild forms of disease versus severe. So we can actually map out if these genes are related to certain functions that are leading to this sort of dysregulation only in the severe forms of disease. So in order to do that, once you have the DGA analysis and your list of differentially expressed genes, we can then go into the pathway analysis feature. And this is where you'll be able to actually see the enrichment result for pathways that are specific only to the severe arginase 1 positive neutrophils compared to the mild forms of disease. And some interesting sort of results from here. So this might take sort of a few seconds to load, but I can adjust the FDR score. So what can we see from the severe population? So we can see that the severe arginase 1 neutrophils particularly have the enrichment of certain pathways relating to both IL-18 signaling pathway, TGF-beta signaling, farcinoid X receptor pathways, and I'll go into how these relate into severe forms of disease. Whereas when we go into the mild group, we can see if we change, again, the FDR score, these particular sort of mild forms or these samples or cells particularly have different pathways relating to sort of metabolism. So it's very different where we see in the severe where it's more inflammatory related pathways, where in mild, it's mainly how the system modulates or how the metabolism is sort of modulated in the system. So how can we visualize even further, or the different sort of key genes in these pathways? So in mild forms of disease, we do see this enriched Krebs cycle disorder pathway, and this relates to its ability to control immune response in mild cases. And this not undergoes any neutrophil hyperactivation. So this is sort of one key concept that we were able to gather as well. We also see that these mild COVID-19 patients also have sort of an enrichment in this phosphate metabolism sort of pathway, which sort of shows us that there's a more preserved redox balance in mild cases, which allows these neutrophils to maintain immune homeostasis, so they don't go into the pathway of sort of excessive inflammation. Whereas if we really go into COVID-19 or severe COVID-19 and fatal COVID-19, we can see a hyperactivation of IL-18 signaling, which does contribute to this understanding of the cytokine storm and how these neutrophils fail to control inflammation. We can also see that in the context of TGF-beta signaling, which was another sort of pathway in the list, it shows that this pathway also aligns with the sort of inability of these neutrophils to prevent controlling that inflammation. And this leads to this worsening outcome, which is the sort of severity of the disease. Okay, so this is just sort of a summary of these pathways that we were able to really distinguish from these specific arginase positive subset neutrophils across mild and disease, severe forms of disease. So this is just a way so you can visualize them further. And sort of what aims, going back to sort of the second aim, so how can we map the spatial distribution dysfunction of these neutrophils expressing arginase 1 in the lung? So for this data set, you can always go into the bulk spatial as well in the Talk to Data system and explore these sort of spatial transcriptomics data sets that would have a lot more information relating to morphology of the tissue and actually querying specific sort of gene markers. So in this sort of geomics data set, we can add our gene, arginase 1, and we can see how arginase 1 is expressed across different sort of layers of the tissue and actually look at different sort of morphology regions as well. So you'll have all of this sort of morphology regions where you can look at the expression of the gene, and you can actually subset only those regions that have a really high expression of arginase 1. So what can we tell from the spatial data, like different results that we can gather? So if we look at the spatial localization of neutrophils with this impaired arginase 1 expression, what we can see is that it's actually different across different regions of the lung. So we're able to actually map using the composition chart where we can see that arginase 1 is particularly sort of co-expressed or regulated with other inflammatory cytokine markers, particularly only in the alveolar morphology region, so not the bronchular epithelium region of the lung. And we can see this actually even further since we are not really working with single cell resolution spatial data, we can still gather from clinical data parameters within the study where the highest sort of level of neutrophil counts in the blood is related to the sort of expression of these sort of cytokine inflammatory markers and this arginase 1 expression. So we can actually also see that in the alveolar morphology, where it has the highest count of these neutrophils in the blood, we can see that it has a higher enrichment of these other inflammatory cytokine markers, which is giving us some indication that there's some dysfunctional neutrophils within that region. So this is, again, some insights that you can gather from spatial data where you're trying to really relate what you're seeing in single cell RNA-seq and applying it to spatial imaging data. Okay, so this concludes sort of the, I guess, a case study for today. I will go through the Q&A just in case there's any questions from the sort of group or clarifications that you need. So I can see that there's some attendees. So what is the profile or the profile in the new Talk to Data? So what does that mean? Okay, so if we go again into the Talk to Data system, and I think the question relates to the profile in the cell ontology or in the tree ontology of the new beta version. So the profile, what it means is that you can actually visualize pre-existing ontology trees that you've generated yourself. So this would be where you would go into the new ontology and you're actually mapping the ontology as well as creating specific customizations to the ontology so that it doesn't sort of use our default or our BT sort of recommended ontology system. So that's what it means is that in your profile, you can actually look and manage different ontology trees. And whenever you go into the actual categories, that's where it'll ask you what specific ontology mapping you will be using and applying sort of the expression analysis information from. So there might be specific cell types that you might annotate in a certain way. You might have already sort of discovered these cells internally and you want to create and map your own ontology for those cells. So you can do that and then look at the expression of certain markers within those cells that you sort of mapped with your own customized ontology system. Okay, so will the new T2D available for the mouse database? So if you're new to Biotrain, you'll notice that the Talk to Data system, as like the former version, includes data sets from not only human, but also mouse and non-human primate data. So we are working on migrating a lot of the features from the former version of Talk to Data into this new beta version where you're actually mapping ontologies, and you might also be able to create a custom data set from there, and also applying it from different data sets, not only from human. So yes, that's a good question. So it's in the process of being developed and it will be integrated very soon. So I hope that answers your question. And I'll check if there's any other questions here in the chat. So I don't see any other particular questions. So thank you everyone, I guess, for joining the session. My colleague did circulate a survey, so you can again let us know if you found this session useful and if there's any other topics that you would want us to integrate and sort of present for you so you can see how you can use the Biotrain system for that analysis. So thank you everyone so much for joining the call.